G-protein link receptors are the largest and most diverse group of membrane receptors in eukaryotes. G-proteins have two specific nucleotides, GTP and GDP. The signal molecule links the G-protein link receptor. This allows for the GDP to turn into GTP. Now, the GTP and the G-protein start moving towards the membrane-bound enzyme. This bounding starts cellular respiration to occur. Since the cellular respiration already happened now, it allows the GDP of the G-protein to lose a phosphate, and it turns back into GDP. Now the signal cell begins to leave, and the G-protein receptor causes the G-protein to move back to the G-protein linked receptor. The signal molecule starts breaking apart and leaving. Tyrosine kinase. These are cell components which are essential in communication. The first stage is reception. The ligand begins to bind to the receptors, which activates the kinase. This causes them to move together and eventually become closed. The second stage is transduction. In this stage, each tyrosine is able to phosphorylate by ATP. As a result, 6 phosphates are come out of these receptors. ATP turns back into ADP. The active relay proteins now attach to the active relay proteins. This process can occur multiple times before a cellular response is activated. The third step is cellular response. In this stage, an activated protein will enter through the nuclear membrane and will activate the transcriptor protein. The protein will attach to the DNA and transcribe a response which is called mRNA. Ligand-gated ion channels are protein receptors with a binding site of a small molecule. The ligand starts moving in towards the closed ion channel. The ligand binds to the closed ion channel and begins to open. The ions now start to move towards the cell, and the ions are equally distributed in half. The ligand leaves and the open receptor